Dear students, welcome to today's live stream. Today we are going to discuss 10 MCQs on molecular techniques. Let's begin the session. First of all, tell me, can you all hear me? Am I audible? Can you hear me? Hi Akash, how are you doing? Am I audible? Tell me. Okay, fine. Thanks for confirming, Akash. So let's try this. Which of the following is the true statement about restriction enzyme? Tell me which is the true statement. They are produced by viruses, cut by single stranded DNA, cut only single stranded DNA, can cut DNA anywhere, restricts the expression of viral DNA. So tell me which is the correct answer. Try this. Okay, so Akash, you are thinking that answer is B. Can you revise your answer, Akash? Excellent, excellent. So you know that restriction enzymes are produced by bacteria. They are not produced by virus. Good. They cut double-stranded DNA. Okay, so they act on double-stranded DNA. Okay, so the viral genome, you know about the viral genome, right? Viral genome can be RNA or DNA. Okay, so most RNA viruses are single stranded and you have very rarely double stranded RNA virus. So similar to that, most DNA, they are all double stranded DNA and you have very rarely single stranded DNA virus, right? And it can cut DNA anywhere. So that is a wrong statement. Why is this a wrong statement? It's going to cut only on particular select sequences. Okay, so they have a specific sites where they will be able to cut the DNA. So all these options are all wrong. So the correct option is restricts the expression of viral DNA. So that is why they are actually known as restriction endonucleases. So Akash, excellent. So their site of action is usually palindromic in nature. Okay, usually it's palindromic in nature. So now let's take up more question on restriction enzyme so you will understand better. Answer this. So try this one. How does restriction enzymes protect against viral infection? Try this one. So Akash, what is your answer? You are saying it is D. What about others who are all in the live class? Tell me guys. Who are all here first of all? Only Akash is answering. What about others? Others also, please answer. So good. So Akash, you are correct that restriction enzyme protect the bacteria against viral infection by cleaving the viral DNA. Okay, so they also methylate the bacterial DNA. That is true only, but that is to protect its own DNA. Clear? So it is to protect from the self destruction. So based on this, there was a MCQ. So that MCQ is like, if a bacteria lacks this methylase enzymes, what's going to happen? So that bacteria will die. So if a bacteria has only a restriction endonuclease but not methylase, it's going to die. So that is why this restriction endonuclease and methylases, they are together. So this is also known as RMS, restriction modification system. Okay, so this is actually known as restriction modification system. This whole system is known as this restriction modification system, right? Okay, so this is fine. Let's move to the next question. Before that, let me introduce about this NEET PG T20. So guys, so we have pre-test series for NEET PG 2021 going on. So this is a mixed bags of 20 clinical questions available, and there are free live tests daily every day at 8 p.m. So guys, you utilize this opportunity. Let's move ahead. This is again a previous question. So here I have mixed previous question and new question, so that you will have both, na? So previous questions are there and new questions are also there. Now try this previous question. Pin 3 is a restriction endonuclease. Which of the following is most likely to be the recognition sequence for this enzyme? So try this. AAG, AAG, AAGA, GA, AAGCTT, AAGGA. Which one? Try this. Which is the answer? Tell me, tell me, tell me. If you see AG, AAGA. So this sequence is not a palindromic sequence. So how will you write a palindromic sequence? So Akash, so already you told me, right? So restriction enzymes, they mostly act in the palindromic sequence. So how to write the palindromic sequence? Let me tell you, it's very, very simple. Look at this. First write any sequence, okay? Then you go here, complementary here in the same strand, okay? So then it will be like, which is the complementary sequence for A? Complementary sequence for A. What is the complementary sequence for A? Excellent. Now you go like this. Clear? Now if you write this sequence, look. Excellent, excellent. Nice, Akash. Can you see? Now, can you understand what is a palindromic sequence? Clear? So this question has been asked in the recent NEET UG. Okay, in NEET UG, 2020. Are you aware of this? So in the NEET UG, they are asking this kind of question. So the standard of question is increasing day by day. So all of us need to update ourselves. Okay. So GAT C. So eco R1. So eco R1. The restriction endonuclease eco R1. Some people pronounce this as eco RI. It's not I, it is 1. Okay, the Roman numeral 1. Right, now try this one. So someone has already answered Parinay Bharadwaj. So Parinay Bharadwaj is saying that the answer is option B. So Akash, now what do you think? What is the answer? What will you say now? Tell me. Is the answer? Look at this. Now can you try Akash? ATTATGC. Can you see this? So it's going to be palindrome A G C T E. Clear? Excellent. So option C is going to be a palindromic sequence. So a palindrome sequence means you should consider both the strand. Okay, it should be like both the strand. Okay, don't think that this is a palindrome AAG. Then now it's GAA. Don't think this is a palindrome. So it's like it's going to act on double stranded DNA. It's going to act on both the strands. Okay, good. So Bharadwaj, excellent, nice. Akash, good. Now let's move into the next question. Which of the following is absent in clinofragment? Clinofragment, before the advent of Taq polymerase, was used in polymerase chain reaction. So Akash, you are asking, is there any specific site for hint 3? So this is the specific site for hint 3, but the purpose of this question is not to test your knowledge on the memorization. There is no need to memorize all these things. Okay, Akash, no need to memorize this restriction sequence of all the enzyme. This question is testing your ability to recognize the palindromic sequence. Okay, so all the other sequence, they are not palindromic, right? Out of the 4 options, there is only one palindromic sequence. That is why it is the answer, right? So no need to memorize each and every restriction sequence for each one. Ripple, Akash, Pankaj, good. So you are all saying, each one is saying each one. Tell me the correct answer. So first, let me introduce the prokaryotic polymerases, DNA polymerases. In prokaryotes, you have 3 types of DNA polymerase. 1, 2, 3. So all these 3 polymerases, they have 2 activities. 5' to 3' polymerase activity and 3' to 5' exonuclease activity. Okay, so all of them have these 2 activity. So for any polymerase, it should have 5' to 3' polymerase. Ripple, any polymerase, it should have this activity. Okay, 5' to 3' polymerase. Without this activity, how will you call this as a polymerase? Right? 3' to 5' exonuclease activity is the proofreading activity. This is a proofreading activity. So whenever this DNA polymerase incorporate a wrong nucleotide, so that can be removed. by this proofreading activity. So all these three polymerases, they have two activity. Okay, only DNA polymerase 1 has extra activity. In addition to these two activities, it has 5' to 3' exonuclease activity also. Clear? So Clino is a scientist. What he did is, look at this. So he took the DNA polymerase 1. Okay, so just assume that this has a 5' to 3' polymerase activity. So this is 3' to 5' exonuclease activity, and this is your 5' to 3' exonuclease activity. What is the role of this 5' to 3' exonuclease? Excellent, Akash. So it is involved in DNA repair, right? So this is involved in DNA repair. So what he did is, he did a limited proteolysis. That means you cleave this protein, and you get a fragment of this, only these two fragment having these two activity. Clear? Subtilisin, Akash. Okay, so this is the one. Okay, so Clino fragment will contain these two, it won't contain this small one. So this will be lost during the proteolysis. Clear? So this is all about Clino fragment. Let's now move ahead. So this one is not there in this. So before taking more questions, we have questions. So before taking this, let me introduce about iconic subscription of Unacademy. As you know that Prep Ladder is now a part of Unacademy. So guys who have already taken Unacademy Plus subscription can go for this iconic subscription. Okay, so those who have this, you will have the following features. So look at this. In the Prep Ladder app, you have recorded video lectures. So in Unacademy, we take live classes. In the Prep Ladder app, you will have recorded video lectures. You will have question banks, rapid revision, and written course. Everything you will get. So you can think about updating your Plus subscription into iconic subscription. You can use this referral code for 10% immediate discount. So with this, now let's move ahead to the next question. So the question may look lengthier. Listen carefully. Which of the following is the correct sequence of the procedure followed in Southern blot? Southern blot was actually discovered by a scientist named Southern. Okay, so Southern was the name of the scientist. After that, they just gave name like Northern blot, Western blot, Eastern blot, so and so. Okay, so you have Southwestern blot, like that. Excellent, Akash. Nice, Edward Southern. So Akash, what do you do? So where are you from? Which college? Parinay Bharatwaj answered us option A. Let's see. Others also try to answer. Okay, you are from Shillong. Nice. Saurav is saying option B. Good. Anyone else? Why today everyone is silent? Okay. Okay, you are a first year students. Good. Anuj Pandey, he is saying option A is the answer. Okay. So those who have not joined our Telegram group, feel free to join the Telegram group. The name of the Telegram group is Biochemistry with Karthi. Also join the Unacademy NEET PG group. Okay, so you will have Unacademy NEET PG group. So I want you to join these Telegram groups so that you will get the instant notification regarding any classes. So before I take classes, I always put an announcement in the Telegram group. Okay, so feel free to join the Telegram groups. So now let me tell you about the blotting techniques. So this is very important to understand the blotting techniques. So in any blotting technique, whether it is Western, Southern or Northern, first you have to do what? First you have to do electrophoresis. There is no doubt in that, right? First you have to do electrophoresis. So from a mixture of protein or nucleic acids, you have to separate this. So if it is Western blot, it will be like you are separating your desired protein. Then what you have to do? After electrophoresis, what is the next step? In that only you guys are having confusion. After electrophoresis, what will you do? So all these blotting techniques, it involves hybridization, right? So electrophoresis is done usually in this agarose or polyacrylamide, right? So it's done on usually the gel, usually done on the gel one. After that, you have to transfer either the protein or nucleic acid into a membrane. So that membrane can be a nitrocellulose membrane, or there is a new one, PVDF membrane. So in our lab we use PVDF. Okay, so you can use nitrocellulose, PVDF. Those days they use nylon membrane also. So first you blot it. Okay, so this is blotting, right? You are transferring. You know what is blotting, right? So for example, in your book there is ink spilled. What will you do? You will take a blotting paper and put that paper over the ink. So the paper will take up all the ink, right? This is called blotting, right? So blotting is you are transferring your protein. Then only you will do the hybridization. Okay, clear? So guys who got confused, I can understand that option A and option B both are slightly confusing only. So those who got confused in the A and between A and B, look at this. Electrophoretic separation. Immediately after that, you cannot do hybridization. The gel will be very fragile. So in the gel, you cannot add antibodies and all. You have to first do the transfer and immobilization. So that is when you are transferring and immobilization, that is in the membrane, right? In the membrane, you will transfer your all the electrophoresis. Here I am telling about the Western blot. So you do all the protein, then you will add a specific antibody. So that antibody is going to bind to only a particular protein. Okay, so after that you do further procedure. Clear? So those who got confused between option A and B, Anuj Pandey, now is it clear for you? Saurav, is this fine? Okay, now let's move ahead with the next question. Which of the following is the reason for which ddNTPs are used in Sanger DNA sequencing? Tell me. So Sanger, Frederick Sanger is the one who sequenced both proteins as well as DNA. He got the Nobel Prize twice. Okay, so this is ddNTPs, not dNTPs. So you have to carefully read the question. dNTPs are different, ddNTPs are different. dNTPs means, so these nucleotide triphosphate, so nucleotides are made up of sugar, base and phosphate. So the sugar is ribose sugar in nucleic acids. In DNA, it will be a deoxyribose. So that means 2' OH group. There you won't have oxygen. Okay, so 2' deoxy. What is the meaning of ddNTPs? ddNTP means you won't have 2' as well as 3'. So both 2' and 3' OH group are lacking. Now I guess you will be able to answer this question easily now. So ddNTPs, they do not contain this 2' and 3' hydroxyl group. Now answer this. So both are absent. Now you can understand why they are used in Sanger DNA sequencing. Let me give a clue also. Sanger sequencing is also known as chain termination method. Other name for Sanger sequencing is chain termination method. So excellent, Akash. So as you have correctly told, why ddNTPs are used? Because ddNTPs, see, ddNTPs, they do not have 2' and 3' OH group. DNA polymerase cannot add nucleotide without an existing primer. DNA polymerase always require a primer, whereas RNA polymerase do not require any primer. You know this, right? RNA polymerase can begin the synthesis of nucleic acid from the scratch. DNA polymerase, it requires a primer. So what is that primer? That primer should provide a 3' OH group. 3' OH group. So Akash, that is for the protein sequencing. So don't confuse the protein sequencing and the DNA sequencing. Okay. So this has to provide 3' OH group. So if there is no free 3' OH group, how are you going to add the next nucleotide? Right? So ddNTPs are going to cause the termination of the chain. So they prevent incorporation of the next nucleotide is the answer. Let's take a very simple question. So before ending this class, let's take up very easy questions now. Now I want every one of you to answer this. Hereafter it will be somewhat easy questions only. Others, these are all the easiest question. How to remember this? How will you remember this? In my mnemonics class, I have already mentioned this. So remember dosa, roti and pizza. So people from southern part of our country, they eat dosa. Okay. People in the northern part of the country, they eat roti. Okay. So this is pizza. Okay. Then people in the western world, they take pizza. Otherwise you can say that pizza is from which country? I think Italy, right? So it's from Italy, so it's western world. So by this you can remember. So southern blot DNA, northern blot RNA, western protein. Will be easy, right? So in the exam, if they are asking you which technique will be used to detect the DNA protein interaction, so what is that DNA protein interaction? Now can you just do a guesswork? Based on this information, just do a guesswork. Which technique will help in detecting the DNA protein interaction? Do a guesswork. Arsh, excellent Arsh, nice. All of you are answering nicely. So for DNA, it will be southern blot. For protein, it will be western blot. So the technique to detect the DNA protein interaction is south western blot. Now let me give you a glimpse how the questions in the next pattern will be. Okay, so the next pattern will be more like clinical, right? So if I am asked to design a question based on the southwestern blot, what I will do is I will select a disease. I will select a disease, okay, in which a transcription factor is mutated or something like that. A transcription factor is affected. Okay, so this transcription factor, what it is going to do? Transcription factor is going to bind to DNA. Obviously, transcription factors are proteins. All the transcription factors are proteins, so they are going to bind to the DNA. So binding to the DNA, they will modulate the frequency of transcription. You know this, right? Frequency of transcription is modulated. So I will take a disease in which particular transcription factor is affected. Then I will ask a question like this. So which of the following technique will help to find the interaction between DNA and this transcription factor? Instead of giving DNA protein interaction, I will just give DNA interaction of transcription factor to the gene or DNA. Clear? So now so something like this, the questions will be framed. So basics are always basics. Okay, so the options, answers will be the same, but the questions are going to be different. Clear? So remember this. So Arsh, what are you doing? So what about you? Where are you from? So guys, you can also ask any other doubt if you have. Feel free to ask me any other doubt other than molecular techniques. At the end of the session, I will give you some time to ask any other doubts also. Okay, so it's not like that. What is the point of this live interaction? So the point of live interaction is you can get your other doubts solved. Tomorrow also I have the session. So if you tell your doubt today also, if there is any image needed, I will bring the images tomorrow and I will do that. Okay, so Akash Saha, I will tell about fish very briefly. Okay, any other one, any other doubts? Anyone? Arsh? Who are all there? Anuj, Saurav, Parinay Bharadwaj. Now answer this question. Which of the following is the best method among the given for DNA fingerprinting? What is DNA fingerprinting? So that is to identify a person. So there is a crime scene. So from the crime scene, what do we take? We take one sample from the crime scene. Okay, good. You are from Chennai. Nice. So in which year are you, Arsh? So this is from crime scene you take. Then suspect 1, suspect 2, 3, like that. Okay. And you collect the DNA samples from everywhere. From the crime scene also you extract the DNA and you take this. From the DNA you are going to find out a particular thing. So what is that thing? That's what we are going to see. So now answer this. Okay, Arsh, you are a foreign medical graduate. Good. So recently I am taking a FMG course in the Unacademy. So if you are a plus user, feel free to join that FMGE course. So for especially for FMG, I am taking a 34 hours biochemistry course. So answer this. Which one is that? RFLP PCR for SNPs, RFLP PCR for VNTR, which is the best method they are asking. See, the best method. Short tandem repeat analysis, RFLP followed by southern blot. Which could be this? So Akash is saying option B. Parnay Bharadwaj is saying option A. Revanshi, just now I told mnemonic. Okay. I guess you just joined, so I will repeat it. So south people, they will eat dosa. So south dosa DNA. Okay. In north roti. So north roti RNA. West pizza protein. Clear, Revanshi? Now answer this question. So guys, are you all aware of something called microsatellites? Microsatellites are repetitive DNA elements which are 2 to 6 nucleotides, repeat elements, repeated up to 50 times. Okay, so microsatellites are repetitive DNA. So 2 to 6 nucleotides. So that it can be a dinucleotide, it can be a trinucleotide. Like, okay, so 2 to 6. The size of the repeat will be from the 2 to 6. The repeat size will be up to 50 times. Okay, so the other name for microsatellites is this short tandem repeat. Okay, otherwise simple sequence repeats, SSR also, simple sequence repeats. So these are unique. Okay, so these days we use, ultimately we are going to do PCR for this only. Okay, we are going to do PCR. So Arsh, it is not the, so those days they were telling. So I think the book you are reading must be a old book. So not VNTR. Okay. So these days no one is using VNTR for DNA fingerprinting. For DNA fingerprinting we are using only STR. Okay, very very important. So in US there is something called CODIS. Okay, so CODIS is a database for this DNA fingerprinting database. So here this CODIS has the data for this only, microsatellite repeats only. Okay, so it's good that I put this question. So many of you may be thinking VNTR. So VNTR is not the thing. So it is this only. All of the following enzymes are used in recombinant DNA technology except, answer this. Okay, so we have just one more question. All of the following enzymes are used in recombinant DNA technology except. Excellent, Akash. So obviously you can see that alanine transaminase is not used in recombinant DNA technology. Revanshi, good. Nice. So all the new members who join now, feel free to join the Telegram group Biochemistry with Karthi so that you will get notification regarding the upcoming classes and all. So online transcriptase we are not going to use. But what about alkaline phosphatase? So we actually use shrimp alkaline phosphatase to remove the phosphoryl groups. Okay, so we use this. We use shrimp. From the shrimp we take this alkaline phosphatase. We use it for removing phosphoryl group. You know, right? Nucleic acid sequence, they are always written in 5 prime to 3 prime sequence. So 5 prime end is the end which has phosphoryl group, 3 prime end has the OH. So from the 5 prime end, if you want to remove the phosphoryl group, you will use alkaline phosphatase. Okay, if you want to add this phosphoryl group, you will use kinases. Okay, so there is something called polynucleotide kinase and all, so that which will add nucleotide. So polynucleotide is going to add nucleotide, right? So alanine transaminase we are not going to use. Good. Answer this. Which of the following is necessarily to be present in expression vector? So this question checks your knowledge on expression vector and cloning vector. So expression vector means it's used to express proteins. Let me give a clue. Cloning vector means you just do the multiplication of DNA. Okay, just clone it. So now you will be able to answer this. I made your job easy. Sharma, Akash. So this is the last question. I want every one of you to answer. Okay. So origin of replication, restriction site, selectable marker. So all these has to be present in both. Okay. So without origin of replication, how will you do the replication? So it has to be there in cloning vector as well as expression vector. Restriction site, we always have the restriction side right in the vector, so that we will remove it and join it. Selectable marker like antibiotic resistance genes, and beta galactosidase. So all these things. So you must be aware of something called blue white screening. We can select the cloned fragments using this blue white screening. So this is a discussion for some other day. So but just let me tell you that selectable markers are like antibiotic resistance and beta galactosidase and all. So this has to be there in both. But ribosomal entry site, obvious, right? Ribosomal entry site, it has to be there mainly in your expression vector, not in your cloning vector. Okay, so to produce protein, you have to have this this ribosomal entry site. Okay. So Akash was asking about fluorescent in situ hybridization, FISH. So yes, in situ. So what is the meaning of in situ? So in situ means in the cell itself, in the cell nuclei itself. Clear? So in that is in situ. So the nucleus itself. So nucleus itself is there. Hybridization means you are okay. Sandwich ELISA, Sharma, what is the exact doubt? Okay, I will tell you about, just to give an overview. Okay, its position I will tell you. So H means hybridization. What is hybridization? Hybridization means two things: either the antigen antibody they are going to hybridize, or nucleic acid strands they can hybridize. Okay, so that is fish. So here we are talking about nucleic acid, so it will be always a probe. Okay, so we design a probe which will be complementary to a particular sequence, so it's going to hybridize. So and these probes will be like fluorescent in nature. So this is like your advanced version of karyotyping. Okay, Akash, so this is like karyotyping fish. So in karyotyping you won't be able to find out micro deletion. You cannot find micro deletion, but in fish you will be able to find micro deletion. Okay, so that means the resolution of fish is better. Okay, so using this fish, what do? You will find out these translocations, all these things, right? So what will we do? Cytogenetics. Even karyotyping you can find out translocation, but for micro deletions and all, so we call this as cytogenetics. Okay, so we go for fish. Okay, so sandwich ELISA, someone is asking, so what is there in sandwich ELISA is, let me just tell you about sandwich ELISA. For sandwich ELISA, antigen should have multiple epitopes. Okay, so this is epitope 1, this is epitope 2. So in sandwich ELISA, the antigen is sandwiched between two antibodies. So two different antibodies, that's the thing, two different antibodies. So this antibody is going to recognize different epitope. So this is the very important thing you should know about sandwich ELISA. So if they are asking you, can you detect thyroxine using sandwich ELISA? Oh, Pratik, glad to know. 663. Nice, nice. So which college are you planning, Pratik? So thyroxine is a hapten. So it's a small antigen, right? It won't have multiple epitopes. So thyroxine cannot be detected by GMC Mumbai. Excellent. Congratulations. So thyroxine cannot be detected by sandwich ELISA. Okay. So Arsh, so further doubts you can ask in the Telegram group. So feel free to ask your doubts in the Telegram group, Biochemistry with Karthi. Okay, so ask your doubts. I will try to answer tomorrow. So tomorrow I will see all of you at 7 p.m. Okay, so same time, guys. So we will have 3 classes. So tomorrow, day after tomorrow, and the day after tomorrow. Okay, so 3 continuous days. We will meet on 7 p.m. and we will try to have a wonderful discussion because of the wonderful audiences like you. Okay, guys. See you tomorrow. Bye.